not going to dwell on this slide much because I have shared it previously, except to say that there has been a long journey over the last 4 to 5 years in understanding the scope and developing the direction for biobanking, including the culmination last year to a broad statewide academic and health service engagement group led by Sunil Lakhani in Pathology Queensland that put up to the Queensland Health Investment Review Board in February this year a stage 0 brief to support statewide biobanking going forward, and it's also working nationally with the NCRIS scoping study to advance and coordinate and harmonise biobanking nationally. So David Hanson was asked, you know, where does he see things going in future? This is my little take on where we're headed and how things are changing and where we need to be in sort of 10 years' time. In that we need to be focused on patient-centred care, shared decision-making, and that comes very important when we think about the dynamic consent and putting consent control in the hands, back into the hands of the patients or the participants. Obviously, we are moving towards adoption widespread across all the HHSs in Queensland with IEMR. We're moving more towards health informatics and virtual or telemedicine care. We're developing a lot of different clinical research infrastructure and technologies, including mHealth applications and remote biometrics of patients in their own home. We're facing issues and challenges around research reproducibility. We're seeing shifts in the regulatory systems and how TGA is going to be approving personalised care options and precision medicine indicated for particular biomarkers rather than disease indications. That's already happening. And then translation of research evidence and innovations. And we are talking largely, I know, within BDHP to enhance Queensland's capacity for clinical trials. So biobanking, I think, is core to a lot of what we're trying to do to enhance our research culture and capability in Queensland. I think it needs to be not your traditional standalone, I have a bank of samples in my freezer in my lab, but something that is digitally integrated, visible, interoperable and connected and underpinning a lot of what we're doing, including clinical genomics. We heard Aideen talk about adoption of clinical genomic tests into clinical decision support systems, obviously infectious diseases, health informatics, clinical trials, complex chronic disease management, cancer care, precision medicine. And we often hear a lot about genomics, but actually biospecimens are used for pharmacogenomics, pharmacokinetics, transcriptomics at single cell level as well. The epigenomic control, the proteomics, the translation of the products of those genes, metabolomics, and for development of diagnostic and prognostic biomarkers, understanding the functions of those genes and gene variations, pre-trial and therapeutic or diagnostic candidate verification. All of this biobanking and biospecimen analytics need to be integrated seamlessly with health informatics and participant control via dynamic consent. Ultimately, the goal is to enable rapid collaboration and access to quality biospecimens. And, this is something that Scott will, I know, be keen on as well, to enable the emerging and mid-career researchers as well as our stellar researchers already, to accelerate our ecosystem for innovation from the point of discovery, from the inception of a project with quality systems through to that translational step and the uptake and adoption of innovations back to the clinic. So obviously, with COVID happening this year, I reached out immediately to Sunil and David Patterson and Patrick Harris and Ewan as well, thinking, well, if biobanking is ever going to be stepping up and centralised, then now's the time to work together to make this happen. So that's, in the background over the last 6 months, there's been a lot of work being developed. And where we're at the moment is that we're putting in a research protocol and ethics application to Metro South shortly. Queensland Health Hero with Melissa Hagan and Karen Thompson are going to be supporting a physical biobank in Pathology Queensland, which will be a physical open access COVID biobank. The recruitment pathways will be a project led by Kim Greaves at Sunshine Coast University Hospital, and they are doing contact calling of previously positive patients, so there are over 1,000 in Queensland, giving us access to follow-up samples from people who have previously had COVID. Recruitment from emerging cases in fever clinics, there's not many of them thankfully in Queensland. Also through fever clinics, contacting and getting negative controls, people who have tested negative, and recall cases to get longitudinal follow-up. So in partnership with BDHP, the board's approved a concept design project to look at digitally integrated biobank interfaces that are going to support that physical biobank that Pathology Queensland is building. The purpose of this is focused on COVID for now, but it's really to develop those capabilities, those research infrastructure for biobanking, of which there are 3. The participant interface, the biobanking interface, and the research user interface as well. So the purpose, I guess, is to enable recruitment and dynamic e-consent pathways and health data linkage, to have a really good system for biospecimen inventory management, and to have that as an interface that's searchable and making those biospecimens visible for researcher access. That's going to ultimately increase the use of the biospecimens and then the value and the return on investment of doing this. So this is well aligned and underpinning, I guess, some of the next stages for that statewide biobank investment concept brief, which we have several recommendations to respond to the investment review board sometime soon. So how this might work is that there are multiple points of interaction with the participants, might be in the hospital and health service, might be in fever clinics, might be in the community through private pathology. But ultimately the patient's going to have a specimen collection. Mostly it's a nasal swab. We want to be able to contact and consent those patients. I'm going to show the flow and how that patient journey might work shortly. But basically you're going to get a specimen to the biobank. It needs to be linked with participant data. That might be from within the hospital and health service, from the Pathology Queensland testing, as well as pulling in data from My Health Records or primary healthcare networks, and directly enter data from the patient around their current symptoms, general health status, and so on. Then there will be, the biobank and biospecimen assays. Now, a number of different data sets are going to be derived. So there is a bioinformatic data opportunity there and a data bank, in fact, for secondary research. And that process of linking the data. And I'm going to talk about how that might work.
Just to warn you, Janet, you are at 10 minutes. You have 1 minute.
Really? All right.
1 minute.
I'm going to have to, that's a diagram of the patient consent journey, but I want to focus on that middle section, and we can come back to talk about how dynamic consent works, except to say that it puts the power in the patient to come back, gives them an opportunity to withdraw consent or manage consent or reconsent for further research. So these are the interfaces that the scoping project is going to explore and work out what's needed going forward. So the patient interface, so 2-point authorisation by the patient or participant. It'll be discoverable, self-registration and information, enabling recontact by the researcher for further projects, but also enabling the participant to come back and change their consent status. There'll be an interface for that biobank inventory system for tracking the use and the processing and the storage. So you've got really good quality systems available, making visible for retrievals, dispatch and tracking the residuals of the specimens. But what's important as well is to make that biobank visible to the researchers. So there's a project happening at the moment where we're moving to have REDCap put into Queensland Health demilitarised zone. And that's going to change and increase the access so that people who are outside Queensland Health will be able to actually be authorised to log in and search that research interface to know what's in there, to submit requests for access. And that's going to enable that increased sharing of not just the specimen, but the data, using those findable, interoperable, accessible, reusable systems.
Thanks,
This
Janet.
is the last slide.
Wind up now.
Yeah, that's the last slide. So essentially, it's really that interface between the physical biospecimen, bringing in the history through the consent or other mechanisms to get that environmental conditions, those data outputs, the different bioanalytics, and linkage with the health information. You know, this is something that we're facing, we've heard around the health informatics, and it touches in on those consents and that social license, but we've got to deliver value out of this. And it's got to fit within the current legislative frameworks, and we've got to be able to resource it and keep it sustainable.
Well, so that sounds pretty easy, Janet.
Open
for questions now.
I'm just going to stop sharing so I can see you all again.
So, David Bunker, what do you need from the themes who are represented today to help support BDHP's vision in this space?
Yeah, look, thanks, Scott. I think we've got to have a fairly tightly constrained scope of work. There's a danger that we try and solve, for everything. But I do think in the exercise, when we're effectively gathering the requirements and trying to understand, how those interfaces that Janice described would work, that we do touch base with the themes, especially the ones that are going to be the most obvious, beneficiaries of this solution. The notion for me is to create more assurance for whatever comes beyond this activity and the journey for biobanking in Queensland. I think it is going to be a digitally enabled environment. I think we've got an opportunity now to capture the requirements and inform the business case and reduce some of the fear and uncertainty about investing in biobank, because I felt that is the biggest concern that many people have, is what is the cost to manage this going forward and how is it going to be working? So I hope this piece of work is going to reduce the concerns there.
Thank you, David. Questions for Janet?
Janet, Ranjani, we've got a very similar project that's been initiated from Sydney over the last few years, and I'll just get together and talk to you about that, because I think if we put you in touch with them in New South Wales, because as you know, they've sort of initiated this kind of thing a lot before Queensland did, and so we've been able to get on board with them. But I think if we can get the two systems talking, it may be to everyone's advantage. Yeah,
we're talking to Jennifer Byrne, but they're still using a kind of paper-based system. That'd be great, Ranji. I'll catch up with you offline. Yep. Thank you.
Ewan, what do you see as the potential links with the statewide cancer?
I think, as Janet well knows, I think this is a vital thing for cancer research in the future, because we're so dependent on genomic changes in the somatic environment rather than germline. So it's going to underline lots of research opportunities in the future, and particularly the integration with data is vital. So no, I'm in this all the way.
Aideen, this might be a good opportunity for you to come in. Genomics and genetic data has been mentioned countless times.
Absolutely.
You've got some thoughts here, and particularly in relationship to the biobank discussion.
Yeah, absolutely. I think I was nodding a lot as you were presenting, Janet, and seeing a lot of the parallels. As Ewan so rightly says, we've moved from an era where genetics relates to rare diseases to now where most cancer patients are having some sort of somatic profiling. So the somatic data tends to be less sensitive from an ethics point of view. But I think if we create from the get-go a framework that will take all of those sensitivities into account, then we'll ultimately have a great system. And I think a lot of the things you've suggested, Janet, for biobanking generally can easily incorporate and adopt genetics and genomic data too.
One thing that's not directly in the scoping project, but David and I have been talking about moving forward, it would normally happen in the stage 1 process, the investment concept brief, but is to hold a community engagement piece of work so that we can understand that social license and what the community feels is acceptable, and just to really get that early feedback from community.
I agree completely. And I think in this area, the extent to which you can maximize transparency is only going to increase trust and increase engagement in the long term. So we've done a few genomics forums for people, and some of the questions I could have predicted and some I couldn't have. So I think it's a really good idea to have consumer forums and public forums on this topic.
And I think it also speaks to the discussion we had before about bringing Nick Waddell and her group.
Yes, I've had a chat with Nick, yeah.
Okay, well, look, I think we better wrap it up. Thank you very much, Janet, for a very concise and comprehensive overview of the BDHP Biobank program. I'm conscious of our time, and I'm going to hand back to David Bunker.